Now we are going to discuss about resting membrane potential. So actually what is this resting membrane potential? In the name only you can understand. Resting means when the membrane is in resting position, the potential which is present inside the membrane or inside the cell, this is called as resting membrane potential. So it is present in almost every cell, and remember that the transmembrane potential which is in resting position. So once we'll also check out the action potential to understand this better clearly. So action potential is mainly seen in neurons and muscle cells, smooth muscles, cardiac, skeletal cells. So all these cells mainly have action potential. It is nothing but the fluctuating electrochemical potential. This is expressed mainly by the sodium potassium ATPase genes or pumps. So the cell pumps, you know about the sodium potassium ATPase, it pumps 3 sodium out of the cell and 2 potassium into the cell by utilizing the energy against the concentration gradient. So yeah, ATP is required. So sodium potassium ATPase mainly takes 25% of the cellular energy. So in our cells, total amount of 25% of energy is only taken by sodium potassium is pump itself. So here ATP is broken down into ADP and phosphorus. So this is how against the concentration gradient, so energy is required in the form of ATP. So cations are lost more than gaining because 3 sodium are going out, only 2 potassium are only in. So you can understand here by sodium potassium pump what happens is 3 sodium are pumped out of the cell and only 2 potassium are sent into the cell. So you can see more positivity is going towards outside and negativity is remaining inside because only 2 potassium are inside. So it is creating a negativity inside the cell. So most of the body fluids are electrically neutral, but the cytoplasmic proteins, phosphates are negatively charged. So sodium potassium ATPase makes the cell minus 5 MV only. So only sodium potassium ATPase makes the cell inside minus 5 MV, but you know the resting membrane potential is about minus 90. Then how only this sodium potassium ATPase alone cannot bring that much of resting membrane potential. So you can see sodium potassium ATPase make the cell minus. During its function it makes the cell electronegative and it is so known as electrogenic pump. So it alone can't create the RMP of about minus 90 millivolts. So RMP is a minus 90 millivolts in cardiac cells. So you can see the potassium leaky channels or ungated channels open all the time. This is very important. Here can the name potassium leaky channels. These are open all the time. So actually you can understand now, as we are saying that the cell, inside the cell there is lot of amount of potassium and outside it is very less. So whenever these leaky channels are always open, what happens is the potassium tries to go outside the cell. As the potassium tries to go outside the cell, you can understand now even inside the cell there is lot amount of negativity going to create. So you can see through these channels the potassium moves out along the strong concentration gradient. As the electrogradient is only minus 5, it diffuses out. So here now at present, because of the sodium potassium ATPase pump, inside the cell there is only minus 5 millivolts, only minus 5 negativity. Now as it diffuses out and cell becomes inside more electronegative because of mainly this potassium leaky channels. So don't confuse with the potassium leaky channels and potassium voltage gated channels. There are 2 types of channels. Not only for potassium, even for sodium we have sodium leaky channels and sodium voltage gated channels, and also we have sometimes ligand gated channels. So we'll see all those things clearly to understand this concept. So as continuously the potassium is now moving out, the internal potential of the cell increases. This is known as potassium diffusion potential. So cells become more electronegative. So we are saying this as potassium diffusion potential because this inside now the cell is electronegative mainly because of the potassium is moving out from the potassium leaky channels. As the potassium is moving out from this potassium leaky channels, now inside the cell it is becoming more electronegative. So the reason for this electronegativity inside the cell is now because of potassium. So it is known as potassium diffusion potential. But before you can see the sodium potassium ATPase pump is leading to only minus 5 MV, and this now it is causing more electronegativity. That number also we will check out. So as this process continues, more and more potassium goes out of the cell. At this time comes where the diffusion potential becomes more, where the concentration gradient balance. So this we need to understand, where the diffusion potential becomes more, where the concentration gradient balances. So it comes to equilibrium, no net movement. So this is known as mainly potassium equilibrium potential, or also called as Nernst potential. So this Nernst potential for potassium is about minus 61 millivolts. So this is what, now as this leaky channel is continuously leaking, the potassium is continuously leaking outside, and finally a certain stage comes where this is equal to the outside. At this time it stops. This is creating a neuron's potential. This is about minus 85. So you can see the large amount of this negativity inside the cell is produced by the potassium, because you can see only sodium potassium ATPase creates minus 5 MV, but because of this neuron's potential or potassium equilibrium potential about minus 85 MV, almost now this minus 85 plus 5 together form the minus 90 MV inside the cell. So the cell is more electronegative inside because of letting out of potassium out of the cell. So normally the concentration of potassium inside the cell is about 140 milliosmoles per liter out of the cell of 4 milliosmoles of liter. So outside the potassium concentration is 4 milliosmoles and inside it is about 140 milliosmoles per liter. So sodium is permeable. Now we have completed the potassium story. Now let us see about sodium, what the sodium does to the membrane. So the sodium is mainly, so now let us take one more cell example. So sodium is mainly permeable and it is letting the concentration, and sodium, we know sodium is more outside the cell when compared with inside the cell. So even sodium tries to come inside the cell because of this concentration gradient, and electrical gradients allow it in. So the cell gains positivity because sodium is a positive, and as the sodium is getting more inside the cell, now the cell of course becomes positive. So the sodium continues to come in, and inside the cell it is electrically positive, so it creates the diffusion potential of the sodium. So now it is called diffusion potential of sodium. Before it is called potassium diffusion potential because potassium is going out. Now sodium is coming in and causing a sodium diffusion potential. As the diffusion potential increase more, it will not allow the sodium in later, as same like potassium. As the sodium is coming more inside towards inside, now it reaches equilibrium position that no more sodium is now able to come inside. So as diffusion potential increases more, it will not allow the sodium in later, and it creates a sodium equilibrium potential. So sodium equilibrium potential is about plus 65 millivolts. So the potassium equilibrium potential is minus 85 and the sodium is plus, because as positive ions are getting inside the cell. So the membrane is now said to be depolarized. So a small mnemonics to remember about this repolarization is, you can remember like Dn and Rk. If you remember this, you can easily remember, because Dn means because of sodium, that is whenever the sodium enters the cell, always the cell undergo the process of depolarization, and Rk means here R for repolarization because of potassium. Always potassium causes repolarization and depolarization is always by sodium. So now as we said, it is plus 65 because of sodium is coming inside. So the membrane is depolarized by the sodium and mainly repolarized by the potassium. So this is what this depolarization and repolarization is nothing but causing a action potential. So this is how the action potential is linked with the resting membrane potential. So here you can check out the cell with potassium, it causes negative polarity or potential, and with positiveness it forms a positive. Of course sodium, it forms a plus 65. So the RMP is a potential difference across the cell membrane, that is from minus 70, it may range from minus 70 to minus 90 according to the cell type. So it is mostly related to the cell potassium level, right, and not related to the sodium level. This is very important. So always remember that if anyone asks you the resting membrane potential, it is mainly dependent on the potassium but not on the sodium. So we also have some formula like here E is equal to minus 2.3 RT by ZF and log 10 of Ci by Co. Here Ci means inside of the cell, concentration inside the cell, and Co means concentration outside the cell. So you can see those representing what structures means water. T means temperature, R means gas constant, F means Faraday's constant, Z means ion charge, and Ci is the concentration inside the cell and Co is the concentration outside the cell. So here you can see the minus 2.3 convert the natural log to the log 10. So according to Guyton, this no need to remember this big formula, very simple formula is now according to Guyton, which is given by Guyton. E is equal to minus 68 millivolts by Z of log 10 of Ci by Co. So the normal value of sodium in the blood is about 145 to 135. And example, you can take CO 150, Ci 15. So just substitute the values. Z is equal to 1 as sodium is monovalent. So if you just substitute the values in this formula, and we get finally the 60 millivolts. So for normal cell, actually E is equal to minus 85 millivolts. And the normal value of potassium in blood is about 3.5 to 5.5. So these are very important values for every doctor, they need to remember. So remember that in blood the sodium is about 145 to 135 or 135 to 145, and the normal value of potassium in blood is 3.5 to 5.5. So if this values also changes, they may cause hyperkalemia, hypokalemia, all those conditions. Those are also fatal conditions. We'll also discuss them. So chlorine is more outside of the cell. Yeah, of course, sodium and chlorine, that is, you can remember like our cells are floating in ocean or sea, that is sodium and chloride are more outside the cell. So according to Goldman equation, we get electromotive force is equal to minus 60 of this Ki into pKni. So this formulas you can remember. So blood and interstitial fluid concentration is same, but the intracellular fluid concentration is different, because we know the blood and interstitial fluid are nothing but the extracellular fluid, and intracellular fluid, we know we have a fluid which is present inside the cell. So at minus 85 millivolts or RMP, the sodium channels are closed but the potassium channels are open. So now you can easily understand this, whatever we say about sodium and potassium channels. So the RMP should convert into threshold to intake the sodium. So this comes about the action potential, the threshold, all these things. Actually always remember, whenever you touch someone, for example if I touch my finger, so now sodium, the signal is sent to my brain because the sodium channels are released, and they, sodium is creating the positive sodium channels are open. So what happens, that positive nature inside the cell, and it as I said, N means sodium is causing depolarization. So first depolarization takes place. So as a lot of sodium comes inside of the cell, the potassium also now starts to get out of the cell. So as potassium also start going out of the cell, again the cell undergoes the process of repolarization. So this current of depolarization and create the action potential, maybe towards the nerve or any other thing, and finally reach the info to the brain. So this is how actually this threshold is called, the minimum amount by which this sodium enters into the cell and triggers up to the threshold. Every cell has some threshold value, so that this action potential is continuous. So this is what we need to understand. So hypokalemia means decreased amount of potassium, that is excessive potassium when comes out of the cell, so make the extracellular area negative due to using drugs in kidney which reduce the potassium in extracellular area, and also by hyperpolarized. And hyperkalemia means massive tissue damage may release the massive potassium into the blood, and renal failure may also cause the hyperkalemia, that is urea is increased, creatinine is increased, and also potassium increase. So this urea, creatinine, potassium, all the markers of kidney dysfunction. And potassium diffuse out less, so less negative. So potassium decrease means RMP decreases, the membrane is hypopolarized. And sodium channels have activation and inactivation gate, this is important. So RMP is normal, then activation gates open, but inactivation gate closes, means whenever the cell is in resting membrane potential, then always activation gate is open but inactivation is closed. Even one gate is closed, then the sodium can't come inside. So when both gates are open, then only sodium can come inside. So whenever there is any action potential or stimulus, then only both sodium means inactivation gate and activation gate both are open, so the sodium can easily come inside. So severe hypo and hyperkalemia can kill the patient. So this is very important. Hypernatremia, that is high amount of sodium. RMP is not dependent on the sodium, so we don't take care of it. So only we take up our most priority is given only to potassium, because resting membrane potential mainly depends on our potassium itself. So now let us see about action potentials also. The electrochemical fluctuations in the cell membranes of excitable cell, that is the potential difference across the membrane, is known as action potential. So the neuron and muscle cell RMP convert into the action potential mainly in the neuron and the muscle cell. So inward of sodium can't depolarize, so these are mechanical sodium. Actually remember that as we said, the sodium channels are of two types, like mechanical channels and also voltage gated channels. So remember that only the mechanical sodium channels alone cannot cause the threshold and cannot depolarize the cell. So only voltage gated channels can create an action potential. So there are two sodium channels, one is the voltage gated and other is the mechanical. So the voltage gated channel only can depolarize and it reaches to the plus 65. So the cell now reaches up to the plus 65 plus charge and later close this voltage gated channel. So whenever this closes, again the process of potassium gets out and a repolarization comes in. So later the potassium channels open and lot of potassium goes out and repolarization occurs and it reaches near to the RMP. So and eventually it becomes a more negative than RMP, then it is known as hyperpolarized. Of course, repolarization means up to the RMP. For example, RMP is minus 90 up to the minus 90. But sometimes what happens is more amount of potassium gets out of the cell. If more than RMP, if it get out, then it is known as hyperpolarization, because now the cell is becoming more negative than minus 90. and the sodium of one cell depolarizations the adjacent cells of the membrane. So this wave continues. So even this depolarization also reaches to the next membrane. So the depolarization is followed by repolarization even in the adjacent cell. So this wave of depolarization followed by repolarization is swept over the membrane. That is, now every part of the membrane, for example, you can understand first this piece, only this part of the membrane is only depolarized, and later, so first, for example, only this part of the membrane is depolarized, that is the sodium is coming inside the cell and potassium is getting out. So depolarization, repolarization is there. But what happens is this is sweeping into the next part of the cell, that is again this some part of sodium from here also reaches here and gets inside. So again this process continues. So like this it sweep out all over the membrane. So depolarization is a sodium influx and repolarization means potassium efflux. So this is a very important key point to understand, very important. So depolarization means what? Sodium is getting inside. Repolarization means what? Potassium is getting outside. So the chemical impulse is balanced mainly by the sodium potassium ATPase. So even along with this sodium potassium ATPase is concerned. So what we need to understand the difference between local potential and action potential. Local potential is non-propagating and it is graded about -70 to minus like this, and it has a property of summation. Means, for example, in local potential, if we give a stimulus of for sodium, we give a stimulus of first 5, and next after some time we give 5. This 5 get added and they go up to the threshold, and thus they undergo the process of depolarization, repolarization. But in the case of action potential, it is not like that. It is propagating, it is stereotypical, and there is no summation. Means at a time we need to give a correct stimulus to open the threshold, and next the depolarization takes place, repolarization takes place. So there is no proper process of summation, but here there is process of summation. So the local potential shows the summation, which is very important. And all or none principle, that is mainly shown by the action potential. So action potential shows all or none process. We may get the full action potential or not. So we cannot say like we have some action potential and it is stopped, and we can't say that there is only no potential like that. Means if it is there, then it is complete action potential, that is the process of depolarization, repolarization takes place. And if no means no, means if there is no potential, then no potential, that is in resting membrane potential. So like that. But we can't say that it is half in action potential, that is not possible. So it is nothing but all or none principle. And next, coming to refractive period. Refractive period means absolute refractive period. They don't move how much we try to do. Actually a refractive period means for example, if this sodium and potassium channel reach up to certain level, like if they are completed their action potential and they take some gap of about like standard 48 hours, I think, they take about 48 hours standard time. Means for example, after completing one action potential, that is a wave of depolarization and repolarization, they take gap about 48 hours, and later they start again their another action potential. This refractive period is what is in which we have absolute refractive period, where if you give any kind of stimulus in between this two action potentials, that in this 48 hours, if you give any type of stimulus any time, if we try it many times or so, it will not move. It will just stay in that resting membrane potential only, but only after the 48 hours only again it starts its next action potential. So this is called as refractive period, and the time absolute refractive period means whatever we do it will not move, that is called as absolute refractive period. So once the sodium goes into the inactive stage, they are not activated, that is what we are saying. So the relative period is a hyperpolarized due to the loss of potassium. Relative refractive period. What is relative refractive period means? As we are saying, it is staying in 48 hours, and actually in this relative refractive period more potassium gets out of the cell. So now a cell moves from the RMP to the hyperpolarized, means it is getting more negative than -90, less -90, more negative than -90, that is called as hyperpolarized due to loss of more amount of potassium, that is called as relative refractive period. And sometimes when they are stimulated, they are not producing action potential. Of course, if we stimulate, they will not produce action potential. Next, coming to RMP. So RMP, resting membrane potential, inactivation gate is open and activation gate is closed. And in hyperkalemic patient, most of the sodium channel in activation gate is closed, and there is no depolarization, of course, in hyperkalemic patients. And next, moving to Accommodation phenomena. It is the RMP near to the threshold is stuck in an inactivation state. So accommodation phenomena is RMP near to the threshold. RMP, the resting membrane potential, is near to the threshold and it is stuck in the inactivation site. So for example, you can understand if it is stuck in the threshold, then continuously the action potential should be forming. So what happens in this accommodation phenomena is the muscles are here weak. The muscles get weak because continuously the potential is going on as it is stuck in the threshold. So the muscles are here completely weak. Next, coming to neurotransmitters, mainly enter other neuron and through receptor, sodium or calcium influx continues the repolarization. So it transmits a action potential throughout the neurons. So it is a chain of electrical and chemical potentials. So we need to understand here the electrical and chemical potential in a nerve. So in a nerve, actually what happens is the neurotransmitters enter the other neuron. Here by diagram we'll understand. For example, this is one neuron, this is one neuron. From this neuron to other neuron, mainly neurotransmitters enter through the receptor by sodium and calcium influx and continues the repolarization. So it transmits as action potential throughout the neurons, and it is a chain of electrical and chemical potentials. So from here, actually in this neuron, from this part to this part up to this end, there is action potential. That is mainly action potential means electrical. This is electrical process, but from here to the other neuron, it is a chemical process. So this is how the action potential pass. This is very important phenomena. Within the neuron, it is by action potential. From one neuron to other neuron by chemical. That is very important. And next, myocardial cells have electrical windows, gap junctions that everyone knows. So later, depolarization and undergo action potential. So factors determining the velocity of action potential. Neurons with wide diameter move fast means less resistant. Yeah. For example, neurons have wide diameter. Of course, these action potentials can move fast there because there is less resistance. But neurons with the thin diameter, they move slow because of high resistance. Because very thin means there is high resistance of velocity. So non-myelinated means there are slow conductors. This is important. We know this is a neuron. If it is myelinated, it is like this. Myelinated means non-myelinated means slow conductor, because action potential now need to travel through the whole neuron and enter to the other neuron. But if it is myelinated, you can understand that it can easily jump like this, because in between there is again not myelinated, right? Of course, myelinated part is insulatory, means here action potentials are not allowed. So this is a technique by which you can see here one action potential form. From here it can jump here. Again from here it can jump. So it can miss this middle parts. So because of this missing middle parts, this action potential can rapidly reach our brain and rapidly reach other neurons. So this is a very good technique. But if it is non-myelinated, then it is very slow conductor. So when someone touches, the sodium goes inside, and mainly voltage gated sodium channels open with the RMP of about -50. So it is called as threshold voltage channel. This is very important. Only the voltage gated sodium channels are called threshold voltage gated channels, but not the normal mechanical channels. So -50 is called here the threshold potential in the case here. So the membrane loses the polarity and becomes depolarized because of lot of sodium is in. So next the potassium voltage channel opens and a lot of potassium gets out and undergoes the process of repolarization. So this is how whenever someone person touches us, how the impulses reach up to the brain. So excitable tissue is undergoing the process of both depolarization and repolarization. And remember that lipids are bad conductors of current, right? So the squam cells mainly act as insulators. These are the insulators. Squam cells, they form the myelin. Myelinated neurons are the fast conduction. Why? Because of course these are not conducting, but they can jump like this. This kind of movement is called as saltatory conduction. So fast pain, that is the delta fibers, are myelinated fibers and carry it, but the dull pain which is carried by the non-myelinated fibers. So dull pains are like itching, tickling, sexual sensations. All these are carried slowly to the central nervous system mainly by non-myelinated, because these are slow carrying fibers. And in non-myelinated neurons, the sodium potassium ATPase should work more. Yeah, because in non-myelinated neuron, this process need to take place all over the neuron. So more sodium and potassium ATPase should work. And in myelinated neurons, they maintain the economy because sodium potassium ATPase should work less. Yes. And the squam cells are also present with unmyelinated neurons, just they don't make multiple layers. Yeah, this is very important. Even in unmyelinated neurons, we have squamous cells, but they are not just multi-layered. For example, you can think this is a neuron and this are the squamous cells. If it is like this multi-layered, means it is myelinated. This is a squamous cell, but only one layer. This is unmyelinated, and only like this neurons, there is nothing around it. Then it is called as diseased. We will see that also here. So demyelinated, make the multiple layers. Yeah. Let us see demyelinated in peripheral nervous system. Demyelinated means this is demyelinated, that is no squamous cell only. So here GB syndrome, that is Guillain-Barré's squamous cells are destroyed and the person become maybe paralyzed and mainly respiratory muscles, then even the person may undergo the process of death. So next, coming to demyelinated in central nervous system. Myelination in normally in peripheral nervous system is done by squamous cells, and myelination in central nervous system is mainly done by oligodendrocytes. So here in central nervous system it may cause multiple sclerosis and also Parkinson's disease. So unmyelinated axons are present in groups around the squamous cell. So this is all about a complete idea of about resting membrane potential and also our action potential. And we will also, even though this video is becoming larger, we will also continue our sodium potassium pump and its importance, because this is also under this discussion. So let us start the sodium and potassium pump importance. So these pumps are mainly present in a cell membrane in all animal cells. Yeah, these are mainly transmembrane, right? And the sodium and potassium pump mainly are under primary active transport. Yeah, if you remember. So they need ATP. It's a transmembrane protein which takes out 3 sodium out and brings 2 potassium into the cell. So it also acts as an enzyme because it breaks the ATP into ADP and also phosphate. So it is also known as ion transporter because this sodium and potassium are also ions. So the destruction of cell may produce hyperkalemia and lead to cardiac arrhythmia or death because the intracellular potassium flows into the extracellular sodium fluid. So about 60 to 70% of the energy is used in the central nervous system and 25% in the whole body. So this pumps made of 2 globular proteins, alpha and beta proteins. These are heterodimers, they are not similar. Alpha is longer and beta is smaller, so they are known as heterodimeric proteins, and alpha is 10 pass protein, this is important, and beta is only 1 pass protein. So the real action in the sodium potassium pump is mainly done by our alpha unit. So alpha unit is very important because the real action is done by alpha unit. It's primary active transport and it's electrogenic pump. So beta is used mainly in folding, anchoring, targeting, and also so beta is just like a supporting structure to the alpha. So alpha is mainly used in the transport for sodium, potassium, and also handling ATP, ADP, phosphate, and should have a sodium binding sites internally. So remember that alpha have sodium binding sites internally and potassium binding sites externally, because now sodium potassium pump will flip and it will bring out 3 sodium outside the cell and potassium into the cell. So the potassium binding site will be present externally, and also remember that ATP binding site also will be internally because ATP also is present inside the cell. So should also have an enzyme ATPase domain, and pump works when phosphorylation is done. So it is also known as P-type pump. So remember that sodium potassium pump is also called as P-type pump because phosphorylation is also going to take place. So now let us see this process and understand how this ATP is functioning. So the first function is to bind with the ATP. So you can see here, this first function is to bind with the ATP, and second is the sodium binding domains are open. So even ATP and 3 sodium are attached. You can say now this is internally, right? So occlusion of the channel takes place, that is even ATP 3 sodium occlusion internally. And fourth one, due to ATPase phosphorylation, the gate flips and its outer gate is open. So now even P plus 3 sodium is converted into E2P as it is flipped. Now it is outside, so it is E2 phosphorus and 3 sodium. So now your ATP is converted into ADP and phosphate. So fifth one, in this the sodium is expelled out, of course. So now E2 with phosphorus and 3 sodium are losing. And sixth process is phosphorylation of 2 potassium loading. So now 2 potassium is loading to E2. And seventh one, occlusion of the outer layer. So E2 P1 with 2 potassium and external occluded. So what is this actually external internal occluded? This external occluded means these are the channels like this, right? So now they occluded here and now they'll release inside. If internal occluded means, before we have talked about that, means here they're occluded and they get out the sodium out. Here they get out the potassium in, 2 potassium in, 3 sodium out. So that what about this occlusion. And next Eighth is flipping dephosphorylation. So before we have seen phosphorylation here we are seeing dephosphorylation in the case of potassium. So here E2P undergoes dephosphorylation, again it becomes E1 inside the cell with 2 potassium inside the cell. So ninth is losing the potassium and gaining the ATP. So again it has come to its original place, E1 plus ATP. So losing the potassium inside the cell. So it can alter mainly by dephosphorylation and phosphorylation. Yeah, we have seen dephosphorylation at the case of potassium, and we have seen phosphorylation at the case of sodium or number of pumps. So here thyroid and insulin mainly increase the activity of the pump, and dopamine decrease the activity of the pump. Even aldosterone, thyroid, insulin increase the activity of the pump. Dopamine decrease the activity of pump. So this is all about detail of our resting membrane potential, action potential, and we also discussed at last about sodium potassium pump and its importance.